So today is our pleasure to have our last keynote speaker, Dr. JohnGilmore. And John is a distinguished professor at the Department of Psychiatry at UNC Chapel Hill. John's research is on brain error development and how it contributes to the risk of schizophrenia and other psychiatric disorder. John is leading the UNC early brain development study. His data is very unique, has more than 1,000 subjects from newborn to teenager, and has twins. Okay? And John has numerous papers and grants. So this is what you can get from ChatGPT. What ChatGPT does not know is John is the tallest person in this room. And the second, John is my boss, good boss. And the most important is John is a local hero for the brain early development. He has been here for more than 30 years and covers, the entire spectrum of brain early development. So it's very great to have you here. So today John's going to talk about early child brain development and schizophrenia risk.
Thank you very much. So I'm going to try not to use this, and let me know if it's not carrying. So I am a psychiatrist and kind of came at imaging through psychiatry. And so what I'd like to do is just kind of talk a little bit about schizophrenia and what we know about schizophrenia at this point, and then about what we've learned about early childhood brain development and how that is or isn't related to risk for schizophrenia. So schizophrenia is a heterogeneous, chronic relapsing psychotic disorder, and kind of the key word for today is heterogeneous. It typically has onset in late adolescence, early adulthood, and it's fairly common. About 1% of people will develop schizophrenia at some point in their life. Schizophrenia has heterogeneous symptoms. You can have positive symptoms, hallucinations, delusions. You can have negative symptoms, amotivation, anhedonia, and about 25% of people have this core deficit syndrome in schizophrenia. And then cognitive dysfunction is also very common in people who have schizophrenia. And when you look at how well people do, how well they live their lives, the things that really get in the way are the negative symptoms and the cognitive dysfunction. The positive symptoms we can treat reasonably well with antipsychotics, and people can learn to live with them to some extent, but the cognitive dysfunction and the negative symptoms are the things that really get in the way of people living good lives. One really important thing to know about schizophrenia and all psychiatric disorders is they're incredibly comorbid. So you never really find someone who has schizophrenia, whatever that is. And so depression is very common in people who have schizophrenia, post-traumatic stress disorder, obsessive compulsive disorder, panic disorder, and substance abuse is also incredibly common in people with schizophrenia. So when we say we're studying someone with schizophrenia, we're studying someone with multiple comorbidities as well. And then the course of the illness is very heterogeneous. About 25% of people have poor outcomes long term. As I mentioned, we can get positive symptoms into remission reasonably well in people. So people can have lots of different constellations of symptoms and outcomes. I have patients in my clinic that are married, that are working full time, have kids, have schizophrenia. I also have people that are living in a group home and can barely put a sentence together, and we call that the same illness. So I want to spend just a little bit of time about cognitive functioning because that's what we kind of study in the kids. So cognitive abnormalities, as I mentioned, are very common in schizophrenia, and they're evident in early childhood. Some of the early studies show that kids who go on to develop schizophrenia tend to walk later and talk later and things like that. Deviations from normal cognitive and motor development are present very early in childhood. And then children who go on to develop schizophrenia tend to score lower on cognitive testing as early as 4 years. And there appears to be a decline in cognitive functioning in late childhood and early adolescence prior to the development of frank psychotic symptoms. And then after people develop schizophrenia, their cognitive functioning is pretty stable overall through their lives. So what's wrong in schizophrenia? And these are some of the early imaging studies of schizophrenia. On the top is a CT scan showing enlarged lateral ventricles, which is actually the most consistent abnormality in people who have schizophrenia. And this is an early MRI scan, and you can see the enlarged lateral ventricles. You can also see the widened sulci in small hippocampi kind of down here, which kind of shows that people tend to have reduced gray matter volumes. They tend to have smaller hippocampuses, thalamus, things like that. And then there's also widespread abnormalities when you do diffusion-weighted images in people with schizophrenia. They have widespread white matter abnormalities as well. So in structural imaging, as I mentioned, enlargement of the lateral ventricles, you get volume loss in the hippocampus and in the frontal and temporal cortex. You have fairly widespread gray matter volume reductions. And these volume, what's important in this is many of these abnormalities are present in first episode never medicated patients. And so many of these abnormalities are present before the onset of the illness and before someone gets treatment. As Paul mentioned in his talk, I can't remember if you mentioned it in the UNC talk or here, but there is volume loss over the course of the illness. And it's subtle. It may be related to antipsychotic use. And what's also interesting, though, is you can get volume loss while your symptoms are improving significantly. And the other thing about the volume losses over time is they could be related to nonspecific things like stress and substance abuse and poor nutrition and things like that. And then if we do functional imaging, there are broad abnormalities in circuits involved with executive function, learning and memory, social cognition. You can get this with task-based studies, and then if you look at resting state studies as well, you see widespread abnormalities in resting state networks, especially in default mode networks. And then it's kind of coming out in the last 2 or 3 years. There's been a lot of studies on these shared imaging phenotypes across psychiatric disorders. So psychiatric disorders have overlapping symptoms, they have overlapping cognitive dysfunction, they have overlapping comorbidity, and they have overlapping environmental psychosocial stressors that could impact the structure and the function of the brain. So as I mentioned, there's a lot of interest developing in these shared neuroimaging phenotypes. But again, that kind of makes the specificity of our imaging markers less clear. And then there were a fair number of postmortem studies in people with schizophrenia. I won't go into detail, but this is just one showing the dendritic spines in the cortex in a normal person, and these are people who have schizophrenia. This is synaptophysin staining in a normal control subject and someone with schizophrenia. And what you can see in postmortem studies is reduced synapses, reduced dendritic spines. But if you look, there's no overall loss of neurons. So you have the same number of neurons. It's more how they're connected together. That is the neuropathology in schizophrenia. And these postmortem studies, again, are somewhat confounded because these are usually people who've had this illness for a long time. And it could be confounded by environmental factors or medications. So what do we know about the genetics of schizophrenia? Twin studies estimate the heritability of schizophrenia to be between 50 to 75, 80%, with environmental factors accounting for the rest of the liability. The biggest single risk factor for schizophrenia is having a parent with schizophrenia. If you have a parent with schizophrenia, you're your risk of schizophrenia increases from 1 in 100 to about 1 in 10. And but while the overall contribution of genes for liability of schizophrenia may be large, the contribution of any single gene is very small. And there's a lot of missing heritability. And I just kind of highlight one of the last big GWAS studies in schizophrenia. They found 287 loci with 120 genes, and genome-wide significant SNPs explained about 2.4% of the overall risk. And then if you expand that to this polygenic risk score for schizophrenia, it explains about 7.5% of genetic risk for schizophrenia. So there's a lot of unknowns about where is all this genetic contribution. There are also people who have altered copy numbers, deletions, or duplications. These are rare, but when you do have them, they're highly penetrant, and but they aren't disorder specific. And then it's interesting, one of the early studies looked at polygenic risk, and they estimate that 8,300 genes in the human genome probably contribute to risk for schizophrenia, and that all of these genes account for 32% of the variance in liability. And I think the latest is that there's about 20,000 human genes, and about 75% of them are expressed in the brain. So there's a lot of different ways that genetics can affect how the brain is developing. And just real quickly, there's new studies showing that these polygenic risk scores, a lot of the risk for schizophrenia is actually not in genes that are expressed very much in the brain. They're expressed in the placenta. So some of this genetic risk is probably related to environmental risk or things like that, kind of downstream from how the placenta's functioning during pregnancy. It just makes it more complicated. And then there's also incredible overlap between these polygenic risks for psychiatric illnesses. So schizophrenia, major depression, bipolar illness share a lot of the same genes, and they do run in families together as well. So the challenges then are that while common variants contribute to genetic risk, most genetic risk is unexplained. It's difficult to translate to molecular pathways, and most common loci have regulatory effects and don't code for genes or proteins themselves. And then let's see real quickly, a couple of really interesting preclinical studies that I think kind of help us temper or understand how complicated this all is going to be. There's a study looking at, this is sensory gating in an animal model, and sensory gating is, people with schizophrenia tend not to be able to gate out or extinguish extraneous stimuli. And so this is an animal model of it, and they looked at this gene, which is important for schizophrenia and bipolar illness and diabetes, and they looked at the genetic background of all these different mice, and the phenotype was highly dependent on the genetic background. So this gene could increase your sensory gating, it could be associated with decreasing your sensory gating, depending on what the genetic background of the animal was. And then I think this is a really interesting study as well. This was done in fruit flies, and what they did is a GWAS study of fruit flies looking at 3 phenotypes. Startle, chill coma, and starvation maintenance. And what they found was, they found a lot of genes that were associated with all of these 3 phenotypes. And then they interbred this fruit fly colony, I think for 40 generations, and then did another GWAS study. And what they found were a lot of genes that were associated with these phenotypes, but none of them overlapped with the previous GWAS study. So it tells us that the human gene, or the fruit fly genome, and probably the human genome, is solving problems using different combinations of genes and different pathways. So it makes it really complicated. There's a lot of heterogeneity of environmental risk as well. Prenatal, perinatal risk factors are important for schizophrenia, obstetric complications, maternal infection, malnutrition, and then living in an urban or a deprived residence. Cannabis use, heavy cannabis use in adolescence will increase your risk for developing schizophrenia. Immigrant status and early emotional trauma also increase your risk for having schizophrenia. But again, none of these are specific for schizophrenia. They can contribute to many different neuropsychiatric disorders as well. And so one of the conceptualizations of schizophrenia is that it's a neurodevelopmental hypothesis, and I think that's what we've been operating on during my career. And so that, pregnancy and birth complications, the early neurodevelopmental abnormalities in children, brain abnormalities are present at first episode, and these genes of risk play a role in development and plasticity. So that's why we believe that schizophrenia is a neurodevelopmental disorder. And this is just a schematic showing how, genetics and then all these different risk factors can interact over the course of development to lead to frank psychotic symptoms. And then, so when I was trying to make sense of all of this, this was an editorial I wrote a while ago, that schizophrenia is the end result of a complex interaction between thousands of genes and multiple environmental risk factors, none of which on their own causes schizophrenia. And when Daniel Weinberger first proposed this neurodevelopmental theory of schizophrenia, he kind of went through all the possibilities, and one of the possibilities that he thought was unlikely was that schizophrenia is not the result of a discrete event or illness process at all, but rather the end of a developmental spectrum that for genetic or other reasons, about 0.5% of the population will fall into. So if you think of all the connections and all the neurons and everything in the brain, that's a normal distribution that, people on the tail end of this normal distribution have a brain that's wired to develop schizophrenia. And so I thought, well, in the face of all this complexity, maybe we can find final common pathways in development that show us how a brain gets wired to schizophrenia. And this is a study from NIMH, Jay Deed and Judy Rappaport and group, and they were the first to really study adolescent brain development, and this just shows the thinning of the cortex. This is normal thinning of the cortex through adolescence. And so there's been a lot of interest in this period of life, puberty. Right after puberty is when some of the early symptoms of schizophrenia start to emerge. There's a lot of normal brain development going on where you get pruning and reorganization of networks in this period. And so if you get to a point, if you think of schizophrenia as a disorder of connectivity, subtle disorder of connectivity, and the black line is normal, and these are abnormally wired or connected, you could get there through an abnormal pruning process, or you could get there through this early elaboration of the early connections in the first place. And when I first started, there was a lot of work going on here. No one had really worked in this area at all. And so that's what we did. And yesterday people were talking about how did you get to where you got. And when I first started, I was doing basic science work, postmortem work, and working in animal models of early exposure to infection. But I was always thinking about how can I study this in a clinical setting. And these are my twin daughters, and this is an ultrasound. It's like, oh my God, we can use ultrasound to study the early brain. And they're 30 years old now, so you can see that I did this a long way. And so one thing led to another. We actually started doing ultrasounds, 3D ultrasounds after birth, and then moved into MR imaging. And so the rest of my talk, I'll talk about some of the studies from the early brain development study. which started officially about 20 years ago. And we have scanned over 1,000 newborn babies. We follow them longitudinally. We, at birth, 1, 2, every 2 years after that. And we get assessments of cognitive and behavioral development. And we have normal kids, kids of moms who had schizophrenia or bipolar illness. And we also had a second grant that was part of this to study twins, so that we can look at how the heritability changes through development. And when we first got started, one of the real roadblocks was that the newborn brain looks a lot different from the adult brain, and there was no way to segment it. And I think I spent a year talking with Guido Gehrig about how important this is. He's a computer scientist at UNC at the time. Please develop a way to segment an infant brain, and I think he was one of the first people to develop that. And of course that's been refined over the years since that time. And let's see, where am I? And then and I think you saw some of this yesterday as well. But there's enormous development in the first year of life. The gray matter volume increases about 50%. And you can see here that the cortical rim is kind of thin, and by 1, it's nice and thick. You can see that the white matter is not myelinated, and at age 1, it's pretty myelinated in, except for the frontal regions. And then by age 2, we've got adult patterns of myelination as well. So this first year or 2 of life is just incredibly dynamic, and there's a lot of development in that. And just kind of quantitating that, we get, in our study, we found that total brain volume increased about 100% in the first year. Most of this was driven by cortical gray matter development. White matter did increase much more slowly and progressively through development. And then we've looked at cortical thickness and surface area, and there's a lot of development in the first year of life. This has changed from neonate to 1 here, neonate to 1 here, a little bit less from 1 to 2. And if you kind of look at it this way, it's kind of interesting. This is regional cortical thickness, and this is neonates, and then 1, 2, and adults are in here. And what you can see is there's just enormous expansion of cortical thickness into the first year of life, and not so much after that. What's also interesting is these regional heterogeneity of cortical thickness is apparent at birth and carried on through the rest of life. Surface area is very different. Surface area expands kind of progressively over the first several years of life. So you're getting different things. And then if this is our cohort up to age 6, and again, you can see this rapid development of gray matter in the first year, and then it starts to kind of asymptote where white matter is just slowly kind of increasing, less so than the gray matter. And then what's also interesting is if you look at gray matter volume, and this is at age 6, and this is gray matter volume at birth versus age 6. And what you can see is that by age 1, the correlation is very, very high. So that individual differences in gray matter that might or might not be associated with later development of psychiatric disease is set up in the first year or 2 of life. And then we've looked at the different parcellation regions as well, and there's similar patterns in the smaller regional parcellations. And again, this is just showing cortical thickness change from 0 to 6, and you can see that overall there's not a lot of change from 1 to 6. It's modest. And in most regions, it's actually already starting to decrease. It's already starting to fall away. And then if you look at white matter, if you look at these early diffusion weighted scans, what you can see is that most of the white matter tracks are very evident at birth. They become more apparent with myelination, but it looks like, from our studies and other people's studies, that all of these white matter tracks are already set up, prenatally. And then, this is just looking at FA development or maturation, or what FA measures. Again, it's kind of a lot of change in the first year of life, and then asymptoting off. And this is up to age 6. This is AD and RD, showing that, again, there's this rapid change in the first year or two, and then it's much smaller after that in the different white matter tracks. And this is just a different way of looking at white matter, showing Dionne's group showed that using this myelin water fraction, you get that same pattern, this rapid development in the first year to 2 of life, and then a much more gradual maturation after that. And I'm not going to go into a whole lot of details, but we did collect resting state data in these kids, and it was analyzed by Ray Gao, who was at UNC and now is at Cedars-Sinai in Los Angeles, UCLA. And what you can see is that there's a lot of development of the complexity of the networks early on in the first year or 2 of life. This is an incredibly complicated slide. I won't go into it, but basically what it shows is that these sensory motor networks are already adult-like at birth, and then the higher order networks have a adult-like structure at 2 years of age. And then the default mode network, interestingly, is kind of in the middle of those. There's obviously maturation after age 2, but it's much more subtle. And so I think I kind of talked about that. And so then kind of switching gears, this is the white matter network. What we looked, our first paper, we looked at the hubs of the white matter network, which are the nodes of the network that kind of control information processing or information flow within the network. And what we found was that by age 1, the rank order of the strengths of these nodes in the white matter network was set up. It was what it looks like at age 6, and our preliminary data shows that this is just carried on through the rest of development. So this aspect of the white matter network is set up very, very early. Many of these nodes that are kind of top eigenvector centrality nodes at 1 are top nodes. About 50% of them are top nodes at birth as well. And there have been some studies in premature infants, and many of these nodes are also evident in premature infants. And this just shows the development or the maturation of eigenvector centrality, and this is between this centrality from 1 to 2, and then from 1 to 6. And you can see that there's a lot of maturation in the first year, and much less so in the after that. So again, it's kind of this pattern kind of everywhere we look. And this is this hub strength across, this is the 78 nodes in an AAL parcellation, and what you can see is this, that they're highly correlated from 1 to 6. So again, just more evidence that things are set up very, very early on in life. And then Yoonmi Hong in the lab did a study of the relationship of structure and functional networks and how that develops in early childhood. And these are the structural connectome or the white matter connectome, and then the functional connectome. And you can see that it doesn't change that much in the first, from 1 to 6. We didn't have really good quality data for resting state in the neonates, so we couldn't include the neonates. And again, this just shows the development of structure-function coupling. And what you can see is that there is some development, but the main patterns are pretty much set up at birth. And then we did use the the Human Connectome Project to compare it to adults, and you can see that by age 6, it's already very similar to the pattern that you see in adults. And then UNC-EPIC used a neural network-based deep learning model to try to predict the functional network from the structural network. And you can see that you can do a pretty good job with this. And then what's interesting is, the structural white matter network looks like it matures before the resting state network. And so what we were interested in is, can you predict a later functional network from an early structural network? And the answer is yes, you can. So this is the structural connectome at 1 predicting the functional connectome at 6, and you can predict that. So they're highly related, and I think part of it is because neither of them change a whole lot from 1 to 6, so it's easy. When you're looking for early biomarkers, maybe the structural connectome might be a better early biomarker than the functional network. And then, so when we think about what we've learned about normal development, there's, just summarizing that, there's enormous growth of gray matter in the first 2 years of life. Is this a period of risk for schizophrenia? Perhaps it's already a little bit disconnected and not wired correctly. Is this also, it is probably, this is when the overgrowth in autism happens as well, during this period of rapid development in the first year of life. As I mentioned, major white matter tracts are evident at birth. Resting state functional networks have adult-like form by age 2. White matter connectome is fairly mature by age 1, and this structure function coupling is also mature by age 1 and definitely by age 2. So by age 1 to 2 years, the overall fundamental structural development of the human brain is largely complete. And then again, there's obviously a lot of change after age 2, but I think of it, this is probably more plasticity and learning and things like that on networks that have already been set up and are functioning. And then I think I have time just to show these. And just this general pattern of development, there's very little human post-mortem work in this age group, but what is available does show this same kind of pattern, that by age 2, the complexity of the cortex is about what it is at age 4 and then later in life. And then if you look at, these are basilar dendritic trees in the prefrontal cortex as well. And by age 2, 2.5, the complexity of these dendritic trees are they're indistinguishable from adults. And then you can look at dendritic spine numbers as well, and you see this same pattern. So what we're seeing with imaging is consistent with the data that we are able to get in in postmortem data. It's also interesting that when you look at the human transcriptome in the brain, in the cortex, there's this massive change in the pattern of the transcriptome around the time of birth, and then it kind of plateaus out, after the first year or 2 of life. So this is also kind of consistent with the type of processes that happen in this first year or 2 of life, and then the processes that are happening, after 2. And let's see. So this is just more data. I think I took that slide out. I thought I took that slide out. Yes. Yeah. This is cortical thickness and surface area from 0 to 6, and I showed you some of that already, but again, these are individual differences in gray matter volume, and I think I showed you these slides, didn't I? Did I show you these slides? I think I did. I'm going backwards, that's why. That's why I'm going backwards. So just a couple of studies then to show this one was just published by Rebecca Stevens, is a junior faculty in the lab, and we actually looked at our children at high risk for schizophrenia and looking at their cognitive development and psychopathology in the first 6 years of life. And what we found was, the p-values didn't show up here. We did Mullen scores at 1 and 2, and then we had IQs at 4 and 6. And basically what we found that there was almost a significant deficit in the high-risk kids at age 1. By age 2, it was statistically significant. And then at 4 and 6, it continued. And then we looked at psychopathology. You know, as I mentioned at the beginning, there's a lot of overlap in symptoms and across psychiatric illnesses. Parents who have a child with schizophrenia, they have a high risk for developing schizophrenia, but they also have a high risk for developing depression and bipolar illness and anxiety disorders. And we're seeing that very, very early on in life. By age 4, you know, they're already significantly more anxious. There's attention problems, hyperactivity already, and then by age 6, you just see this broad pan domain increase in psychopathology in the kids that go on to develop, or that are at risk for schizophrenia. And then one of the interesting, you know, is how do they do these cognitive deficits? How do they progress during this time period? And what we found is they don't. So you know, they're set up pretty early. There's no significant change from one. These are like Z-scores from the different assessments. And so the cognitive deficit that we see in people who are at least at risk, and probably in people who have schizophrenia, is there very early, and it seems to be fairly stable through early childhood. So I think I talked about that. And then I just wanted to highlight this paper. This was a somewhat recent paper that we did with Daniel Weinberger. And as I mentioned, the polygenic risk score for schizophrenia includes a lot of genes that are preferentially expressed in the placenta. So we looked at the polygenic risk score for schizophrenia that were placental. It was a placental schizophrenia polygenic risk score in our data set. And what we found was that there was a significant decrease in gray matter volumes in boys at age 1. And then we also found that in boys as well, there was a significant delay of cognitive development using the Mullen scores. And we tested the polygenic risk score for autism, attention deficit disorder, depression, and bipolar, and we didn't see this relationship in any of those. So and we looked at the larger total schizophrenia polygenic risk score that included non-placental genes, and that didn't have this relationship as well. So it looks like maybe some of this interaction, I forgot to mention, I'm sorry, this is an interaction between that polygenic risk score and obstetric complications. So it looks like that may be what's driving some of what we're seeing in schizophrenia. So I think I'm getting, so we're kind of finishing up now. We scanned our last 10-year-old about 3 months ago, and we're trying to lock down the 8 and 10-year-old data set now, and so we'll be able to extend these things into late childhood. And I think what we're really interested in is following these kids through adolescence, and to see where these individual differences in brain structure get set up. Is there more modification, or do more individual differences happen during this second critical period of at least cortical development after puberty? So we're real interested in how those two interact with each other. And then these are just some preliminary results, again showing development out to 10, and this is the relationship of the neonatal scan. This is total gray matter volume at birth to 10, and there's a significant relationship already. I mean, even this far out, and then at age 2, it's much higher. So a lot of these individual differences from early development are being carried out through the rest of childhood. So in summary, psychiatric and schizophrenia and other psychiatric disorders are very heterogeneous and complex, and making them, I think, difficult to study using imaging because of how complex these phenotypes are. We need to develop methods to study the complex relationship between individual differences in brain networks and unique individual phenotypes. Much of the fundamental network architecture of the human brain is in place at ages 1 to 2 years, and these individual differences in these networks also arise in early childhood. So I think as much as these networks are going to be related to phenotypes, we will be able to detect it very early in life. We've done preliminary looks at the relationship, and there are papers that I haven't talked about relating cognitive development to gray matter development and white matter development, and we do get relationships, but they're kind of modest, 0.2, 0.3, and UNC-EPIC is doing machine learning now with the networks and cognitive outcomes at 8, 6, and 8, and we see about the same. It's not giving us a lot of advantages over looking at the traditional things, at least at where things stand at this point. So yeah. So I think we just acknowledge everybody that's worked on this over the years, and so that's it.
Good talk, and I have a couple questions for you, but I will start from the most important one. So I understand this is a longitudinal study. It's a long effort, it's a year long, it's a very long process. So how do you control the confounding variables, for your subject? Because I understand this disease has a genetics, has a contribution, environmental factors has a contribution.
Yes.
So how do you control those confounders?
Yeah. I mean, we do look at things like, the other big confounder in all of this is the scanner, because over the course of 20 years the scanner's changed 3 times, and our sequences, I try not to change the sequences that much, but when they get so much better, you just have to change. And so we've been trying to, we've been harmonizing our data with combat, and I mean, I think the biggest confounder is socioeconomic status, and we definitely control for that in many of these analyses. And when we first started, we didn't, we were just trying to prove you can do scans on kids and you can analyze them and you can follow them in the first couple of years of life. We didn't do a whole lot of childhood adversity stuff, which we have collected in the older kids, but it's more of a retrospective assessment of that. So we have a lot of that data that when we start analyzing our 8 and 10-year-olds, we'll be able to put those in our models as well. But you're right, it's, I think,
it's difficult.
And I think that's the conclusion is like, everybody with schizophrenia probably gets to having schizophrenia more or less in a unique way, and how do you study that, and how is that related? Because I think as the genome solves problems using many different combinations of genes, and you can solve the same problem using different combinations of genes. I bet you the brain does the same thing. That you can have a behavior that is due to many different combinations of wiring and correlations and things like that. So it makes it tough to study.
Any questions from the audience?
I have one question. So how do you define or quantify the structure and the function coupling?
How?
Yeah, how do you couple the structure and the function?
Yeah, it's the. Let me get the paper and I'll tell you. I don't remember off the top of my head. There are different ways to do it, and I can't remember exactly which way we used it. But it's a regional kind of thing. How the coupling within the white matter connectome is related to the coupling in the structural connectome in a certain region. Yeah.
So do you see any difference between genders or between different
We did not.
developmental stages?
We did not see differences in gender.
Okay, thank you.
I was wondering, could you comment on any kind of interventions or things that you think, it seems like it's an early setup, there's multiple pathways of kind of getting there, so it seems kind of almost would be elusive or difficult to sort of intervene on, but could you just comment on anything you sort of see?
That's a good question, and if you can find an early marker, then that's the next question. What do we do about that? And I think the first step is just going to be just anything that optimizes good, robust, resilient brain development will probably impact risk down the line. So just a low stress, environmental enrichment, things like that. I think that's where we would start. Not to ECT right away.
Cool.
John.
Yes.
So a lot of your data were real tight, like year 1 or 2, and it was set, and it went all the way out.
Yeah.
But of course, there's variability.
Yes.
And I'm wondering, since schizophrenia isn't super common, do you think it's those that don't fit this pattern that might be the ones that are actually at risk, and the ones that are tight
Yes,
in the pattern are they're the kind of safe ones?
Yeah, we've looked at that at least to age 6, and we didn't see much of a difference. We got like 2 groups out of our thing, and one was groups with big brains, and one was group with small brains, and the schizophrenia didn't fall into either one of those very specifically. So, yeah, but that's definitely where we want to go. Is this once we have the whole longitudinal trajectory, is that going to be informative or not? Yes.
A super quick question, and John, thanks for such a fabulous overview. It's really nice to hear it from you and showing the lay of the land. You propose a sort of really interesting model where either there's an early hit and that persists, which we've been talking about, or most of it happens at puberty and there's nothing really evident earlier on. And one of the things I've seen in the field is some sort of data-driven subtyping. And so you take these trajectories and you say, look, these people look like they have the early hit, and it strikes me you're in a position to do that right now or very soon. So my question is, what is your opinion of these sort of data-driven subtyping? It strikes me that you could either do subtypes clinically and say, let's go look at the imaging, or kind of the other way around. I mean, do you think that's a fruitful way?
I do. Yes. And then you can see, do my kids fall into one of these subtypes more or less frequently? But I think that's definitely a way to get at the heterogeneity. Yes.
So John, I have a question for the computer scientist here. So you showed the performance by graph neural network.
Mm-hmm.
I think it's remember it's 71%. So do you think it's a good number, or
I think
can we use this in the
from other studies, I think that's pretty good. And I know you were getting, when you did your genetic one.
Yeah. Well, that's a bit different. Yeah,
It's a
I'd
bit
love to
different.
chat about that. It's a bit different.
Yeah. Okay. Yeah. So I think it's pretty good. This isn't my hardcore area, but from what I understand about it, it's pretty good.
John, thanks for the talk. I was very interested in the related so that the earliest structural connectivity can predict the later functional connectivity. So when you check the time scale, I suppose different rate of the development might give a different prediction window. So some kids might have a better prediction if they
Yes.
earlier, if
Yes.
they go faster.
Yes. We haven't broken it down. This was just a group. We haven't broken it down by individuals at this point. But that, yes, to be able to do that kind of thing, I think would be really, really good.
Thank you.
Yes. And that gets at kind of the trajectory as well, just the overall difference in trajectory and how they're related.
Any other questions? Now we just